So today we're going to talk about osmosis, active transport, and then the transport of large substances across the membrane. So to start, let's just review and refresh what we've been talking about so far. So we are in the section of cellular transport, which is when we have to move across the membrane and either go into the cell or exit the cell. And there are two main types of transport. The first one is passive transport. So this requires no extra energy. This would be diffusion and osmosis. And then our second type of cellular transport is active, which does require extra energy. So today we'll talk about osmosis and then active transport. So osmosis is the diffusion of water across a semi-permeable membrane. And so that water is going to go to the higher solute concentration. All right, so I'm going to write water moves to the higher solute concentration. And it's going to keep moving until it equalizes the two concentrations. Okay, so let's talk about what does this mean? Okay, so water moves to the higher solute concentration. So the solute are these like items in the liquid. Okay, the liquid here, it's our water. We call that the solvent. Okay, so let's add that here to our notes. So water equals solvent. Maybe particles in water equals solute. So during osmosis, water is going to move to the side that has the more solute. So if you look at this picture here, the left side has just a few of these purple particles. This side has a bunch. So this side has the higher concentration, which means that water is going to move from the left to the right side. And it does that because it wants to even out the concentrations on both sides of this. So we want the same concentration on the left as we do the right. So this is what happens over here. We get more water on this side. So now we're evening out the concentration on both sides. All right, on this slide, I have a YouTube video that shows you more how osmosis works. On this recording program, I can't play the YouTube video, so I will post the link to this on our website. Be sure to click the link. Again, it just shows you how osmosis works and how you know which direction the water is going to go. But if we look at this picture here, we see that on this side, it looks like we just have water. And this side, we have some water, and then the green substances here, this is our solute. So this side has a higher concentration. So that means that the water is going to move through the membrane and into that side. All right, so when we think about the cells, we think about the cell in the terms of its solution on the inside of the cell and then the outside of the cell. So here we have this is our cell. Okay. So when we talk about inside, we mean all the solution inside the cell. And then when we talk about outside, that's any solution or liquid surrounding the cell in your body. Okay. And so there are three different types of solutions that your cells can be exposed to. So one of them is isotonic, hypertonic, and hypotonic. So the first type of solution is called isotonic. Iso means the same. All right, so ISO, this means the same. And so what that means is that the solution of like the water and the solute is the same on the inside of the cell as it is on the outside of the cell. So if you look at this picture here, inside of the cell, we have a 10% salt solution, and then outside of the cell, we also have a 10% salt solution. Okay? So both are equal. Our inside and our outside are equal. We also call this equilibrium. Okay, so when we're in an isotonic solution, we are at equilibrium. However, remember when we talked about equilibrium and diffusion? Just because you're at equilibrium doesn't mean that those particles no longer move. Our water molecules will still move in and out of the cell through the membrane. It's just for every like one water molecule that would move in, one would go out. So our net movement is 0. All right, our second type of solution is called a hypotonic solution. Hypo means low. So think hypo is low. So what this means is that the cell is in a solution that has a lower concentration of the solute. Okay, so if you look at our picture here, inside the cell, we have a 20% salt solution. Outside the cell, we have a 10% salt solution. Okay, so when we say that it's in a solution lower, outside the cell has less solute particles. Okay. So that means water is going to want to move into the cell to dilute this salt concentration so it becomes equal at 10% on the outside and 10% on the inside. So you need to know that the net movement of water is going to go through the membrane and into the cell. All right, so let's talk about what happens in a hypotonic solution. Okay. So when a cell is in a hypotonic solution or a hypotonic environment, that means that water is moving from the outside and into the inside of the cell. This is the exact same thing that happened when we did our argument-driven inquiry lab last week. So you had the starch solution inside your cell, and then you had the distilled water on the outside. Because that starch solution had way more solute, water moved from the outside into the cell. And as we saw last week with our data, that it got bigger, right? Our mass increased. I noticed some groups, they were even able to tell that the size of their cell models got larger. And so in animal cells, if they're in a hypotonic solution, water will keep moving into that cell until it reaches equilibrium, or it can cause the cell to expand so much it can even burst. Plant cells, the same thing happens where water moves in, except they have that rigid cell wall. Therefore, they don't burst. They just become more firm. And this is a technique that your grocery stores use. How many of you have gone into like the produce aisle at the grocery store and you see like the sprinklers and it actually waters the produce? So grocery stores do this on purpose to create a hypotonic environment. So the water that they spray on will move into their produce, and it causes them to become firm. And of course, that's more desirable to purchase. Our third type of solution is called a hypertonic solution. Hyper means higher. Okay, so think hyper, higher. So this means that the cell is in a solution that has a higher concentration than the inside of the cell. So if you look at this environment, on the outside now, there's a 20% salt solution, whereas in the inside of the cell, there's only a 10% salt solution. So the higher concentration is on the outside of the cell. And so water is always going to move towards that higher concentration. Therefore, water is going to move from the inside of the cell to the outside of the cell. So on this slide, you need to know that the net movement of water is out of the cell. Okay, not met, do net. Net movement of water is out of the cell. So when this happens in our animal cells, it just causes them to shrivel. So there's less pressure to hold that cell in its original shape. So it shrivels. This happens to us, like if we've been in the water for too long. If you've ever wondered why your skin wrinkles, this is why your skin wrinkles. On plant cells or in plant cells, when they're in a hypertonic solution, it causes those plant cells to start wilting. Okay, so that's it for osmosis. So real quick, I just want to summarize. So remember, diffusion is the movement from high concentration to low concentration. And then osmosis is the movement of water, and it basically is from the lower concentration to the higher. So water goes from low to high. So these are our two forms of passive transport, meaning that these don't require any extra energy. So now I want to talk about active transport. So active transport is going to require extra energy, and it's still transport though. So we're still talking about the movement of substances across the plasma membrane. Okay. And these are going to move against a concentration gradient, which means that they are going to move from a low concentration to a high concentration. Okay, so for this slide, you need to know active transport requires energy. And you need to know it's when substances move from a low concentration to a high concentration across that plasma membrane. So if you take a look at the diagram down here, we're going to move in this direction. So here I have just two of these blue particles. Over here I have a lot more than two. So this is my high concentration. This is my low concentration. So to move through this channel, I need energy to make that happen. ATP is energy. In order for active transport to happen inside your cells, you have to have a carrier protein. So you have to have one of these channels in your membrane that allows certain substances to go through. Since these require extra energy, they're called a pump. You have a variety of different pumps in your cell membranes. I'm just going to point out one example because it's probably the most popular one. One example is the sodium potassium ATPase pump. So this one, it's complicated how it works, and so we don't have to know the entire mechanism of how it works, but it is responsible for keeping your sodium ion levels and your potassium ion levels maintained within your body. So what you really need to know for active transport is just that this type of transport requires energy, and it moves from low to high concentration. All right, so now I want you to take a minute, and I want you to see if you can answer this question on your own. Why does active transport require energy while passive transport does not? So take a look at these diagrams and see if you can come up with a good answer. All right, so if you thought about the answer to this question, hopefully you thought about a couple things. So first of all, we have diffusion over here, just simple diffusion. These substances, they can just freely pass through the membrane. They don't need anything extra like a carrier protein. And they're also moving from high concentration to low concentration. Next, we have facilitated diffusion. So for one, these are both moving from high concentration to low concentration. But also, if you look at how they fit in those unique channels or those protein channels, they also fit, right? Like it looks like it fits nicely. Like call back to the bubble membrane lab we did and we put the rubber band on the bubble membranes. If you just drop something that fit through the rubber band, it would just easily go through. You wouldn't need any extra energy to make that happen. And then finally, for the active transport, if you look here, so we have different substances that need to get through. They might not fit quite right. Like if you look at the shape here, the shape doesn't fit. Also, active transport is going to move from low to high. Now, the last thing I want to talk about in regards to cellular transport is the transport of large particles. Okay, so how do we get large molecules either into your cells or out of your cells? Okay. And so in order for these to move into your cells and out of your cells, they have to be packaged. Okay, so large particles, they must be packaged in membrane-bound sacs. Okay. So kind of similar to like our big old cargo like shipping container. Okay, you have to package your large stuff. When we talk about the transport of large particles, we break it up into two different mechanisms. So one of those mechanisms is large particles exiting the cell, and then the other mechanism is large particles entering the cell. So the first one we're going to talk about is exocytosis. Exo means exterior, okay, or you can think of like exiting. So exocytosis occurs when you have large particles that need to leave the cell. So what happens is the Golgi apparatus, it's going to package them into their membrane-bound sacs, which are called vesicles. And then these vesicles are going to fuse with the membrane and end up releasing the contents. Okay. So here, these green dots, so these are our large particles. These are like waste. Our cells don't need them anymore, so we have to get rid of them. So the Golgi apparatus, it packages them up in these vesicles. And the vesicle moves until it gets to the membrane. And then it kind of just fuses to the membrane. Okay, so there's not an opening of like your front door wide open where anything can go in or out. It doesn't work like that. So it has to fuse to the membrane. Once it's fused, again, it closes off so nothing can just freely go in or out of the cell. And then finally, it can open up into, or the membrane cuts off. So if you look, so like the membrane will close itself off like it here and here, and it'll open up. And then all of your waste particles can freely exit the cell, while all at the same time, your cell membrane is not impacted. Okay, so there's still no opening between the exterior environment and then the interior environment. The second type of mechanism for the transport of large particles is how do we get large particles into the cell? So this is called endocytosis. Endo is going to be inside. Okay. So in order to get large substances inside the cell is going to surround the large substance and kind of enclose part of it into the plasma membrane. Then it's going to pinch off and leave the substance inside the cell. So if you take a look here, so here are large substances that we're trying to get into the cell. So the membrane is going to move up and fold around it. Okay. So it's going to make a full closing. and then it'll be able to pinch off. So this part of the membrane will still pinch together, and it'll keep the membrane intact. So again, there's no opening between that outside environment and the inside environment. And then you have the large particle surrounded by the membrane. And then eventually it gets like dissolved within the cell, and then whatever particles you're bringing in, they are freely able to move throughout the cell. All right, this last slide, it's a picture of amoeba ingesting large particles. So you can see this process of endocytosis. Again, on this recording software, I can't play the YouTube video, but I'll post the link so you can take a look at what this looks like.